Great. Cool. Rachel, I think you're muted, just so you know.
I am muted. Yep. Classic. Sharing entire screen this time. Okay, cool. All right. So hi, everyone. I'm Rachel, and I work with Julian and Ajith, who you know better than me. So I'm going to pass it over to them to kick us off. And everyone should be able to see the screen that I'm presenting.
Yes.
Everyone sees the slides?
Yeah.
Great.
Cool. So, hello, everyone.
Good morning.
I'm Julian. I am one of the technicians in the Meyer lab, Rachel's lab, and one of the co-managers of the CIB project in the lab, both for logistics and actual lab work itself.
Hi, everyone. I'm Ajith. Nice to meet some of the new faces. And yeah, I'm one of the lab techs working on this project. And yeah, we've been receiving and processing all of the soil samples for this project. And we've made some great progress and have some things to share with you guys today. So really excited for that.
Yeah. So yeah, what is eDNA Explorer? eDNA Explorer is a kind of a platform to put your environmental DNA data. It's a way to view and kind of share data across different communities. Anyone can access it, and anyone can upload to it, which is really exciting. Just as a community science platform, similar to like iNaturalist, where you're able to share data across public settings. And yeah, it's the platform that we use to upload all of our eDNA data, and it makes it accessible for people like you to access our data.
It's also the web engine that the compute engine that processes the raw FASTQ files and assigns taxonomy. We bring in geospatial data automatically. So not only can you manage your project, but you can process it really, really quickly. The results are available in like a day, and we include a lot of uncertainty and accuracy statistics along with everything in your downloads. So the raw ASVs are available through the project pages on the eDNA Explorer. And we'd love to host other people's sample data as well, so that you can look across projects at like patterns of where certain organisms were found. Just jumping in. And originally we had this project where eDNA Explorer was part of CalATBI at UCSC, and we spun it out as a company to be able to maintain it. So that's the partnership between UCSC, CalATBI, and eDNA Explorer. It's sort of one multi-armed, multi-pronged thing. Back over to you guys.
Okay. Yeah, so in terms of CalATBI, we're based here at UC Santa Cruz at the beautiful Coastal Science Campus, and we're a community service lab. We're mostly actually lab technicians, and we specialized research scientists, and yeah, we have around 50,000 samples in our collections, which are mostly public research resources. Some are private. And we pretty much do eDNA processing of all sorts, from DNA metabarcoding to single species qPCR assays. And yeah, we're a very dynamic lab working with a variety of land managers pretty much across California and the country. So it's really exciting. And our website even has protocols available to the public, so people can see our metabarcoding workflow and download protocols as well. And yeah, our website definitely needs updating, but stay tuned for that. And me and Julian are basically the main contacts for this project. So if you guys have any questions about really anything, feel free to contact us. And in terms of CIB samples we have in our current collections, we've received and accessioned around 4,585 soil samples in 5 ml cryovials. And these are stored at minus 80 degrees Celsius, so the DNA will be preserved for a very long time. And we will share these links with all of you guys in this slide presentation. But this link here that says CalATBI CIB Museum Archive, that is our full inventory of CIB samples, in which you guys can go through and query specific samples, and you can see it has your normal sample ID, which will be familiar to you, and we have a processing ID, which are those G codes that we use when we're doing lab work, so that way we don't have to write the full sample ID on every single small tube we're working with. And you can see on the far right, that's who contributed that collection or sample. So you'll see a variety of organizations there.
There's additional metadata in other places, like the other field metadata and the geospatial metadata. So we'll cover that later. Thanks, Ajith.
Yeah, and so from that museum archive, we have processed 576 soil samples with an additional 250 priority samples being processed currently. And those soil samples that we've already processed and have data for are already up on eDNA Explorer, and they're uploaded as plates. So we process samples in batches of around 96. So with blanks, it's around 90 samples. And so they're uploaded onto eDNA Explorer in 7 separate projects. And soon they'll be able to be combined into a single umbrella project, just so then you'll be able to do these sorts of comparisons all in one spot. And it's really helpful for downloading all of your data in one spot. But for now, they're separate. And we have a link down here below that lets you search for your specific samples. And then access which plates they are on, so which eDNA Explorer projects they are on. And just to give a little overview of what we are processing, so we're using 4 different primer sets, metabarcoding markers. The 16S marker, which targets bacteria, and then CO1, which targets invertebrates, and then 2 different fungal primers. I think that we accidentally put in ITS2 plants, but it's
I just
actually
realized that we did. We'll fix that on the slide.
but we have been using 2 different
fungi.
Yeah, ITS2 for fungi, so not plants. But yeah, so Rachel, if you could click that link, I can just show them briefly how that works.
Sure.
Let me see. So you're able to search for your sample. I've cleaned the sample IDs to let you search for your specific sample, excluding our processing ID. And then you can find which plate it's on and click that link. And from that link, it will take you to the eDNA Explorer project page, and you are able to download like ASV data, the raw data, or the processed data from here, and look at all the different functions that eDNA Explorer has. But like, for example, that plate, we had 2,143 species of fungi detected, or I guess not just species, but groups of organisms. And
I'm just going to show them a couple features while we're here.
Great.
So you can see in this project where like anything you click on was detected. And then you can also look at it against all public projects, and a map should show up here. And you can see we have much more data in California than other states. Nobody's surprised that cladosporium is all over the place, but I could have picked a rarer example. And you'll also be able to see the other projects that it was found in that are public and explore those. Yeah, while we're here, you can see the contributors here. We're happy to grow this list. We're going to be adding more features to the eDNA Explorer every couple of weeks now. We are getting functional diversity coming out soon. You can see random forest predictions of the beta diversity. So this is the community composition across the samples in this plate. And then the alpha diversity, there's random forest coming soon for that, but you can see that there's all sorts of different levels of samples. You can toggle across your primers here and see individual models. So here for bacteria, there's really cool soil predictions that are really strong. This model is an R-squared of 0.8. And then for fungi or for invertebrates, let's say you wanted to go to see what's been found in GBIF, you can take that to what's been found in California And you can see the overlap of what's in the CO1 data with what records are in GBIF. There's some bugs here sometimes, like not these types of bugs, but other bugs, like computer bugs, where sometimes things don't show up, but we're really trying to improve this and make it perfect. So you can see that there's a lot of overlap, and sometimes it's really exciting. We're like, hey, this type of longhorn beetle hasn't been recorded, and I don't know if this is exactly correct, since 1983. Sometimes that's like a taxonomy shift naming issue. Maybe this lichen is really rare. It was in 1% of the samples. The last observation in GBIF entered was from 1957. So definitely check that out for leads, and also let us know if you find like errors, okay? That's all I wanted to point out right now. You can look at organism trends and like site patterns for when but this will be much more meaningful when we have the samples all in one umbrella project. Okay, sorry, back over to you.
Yeah, so I think that's it for that slide. And then I can go over this really briefly, but we are currently processing 250 samples that were priority samples. Colleen sent us a list, and we've kind of narrowed down what we're processing. And as of today, we'll be done with PCR. So from here, it's going to get a lot faster. And yeah, we're hoping to have, not hoping, we will have data by the end of March or even sooner. And yeah, that's kind of it for our processing.
Yeah, and then once data is available, we can invite you guys to be contributors, so you guys can download all of that data. And we know for some of the data and for some of the samples that we discussed that some of the locations need to be obscured. So we have two options, and maybe we can kind of have a discussion about it now. So option one is that we have a private project for just that batch of samples. And option two is that we still upload the data and have a public-facing project, but those locations are just obscured to a really coarse resolution where you can't really see the points on a map. So yeah, if you guys have any preferences on how you prefer the data is available, let us know.
I think just putting like your opinion, like your vote in the chat, and this is actually for Point Blue. So it's really their decision to make, but ideas of going forward for other public projects, or how you might want to be able to leverage other people's private data in an ethical way where the locations are not revealed. It'd be really nice to know what you think.
And that's something where we're working on solutions globally across all the ATBI projects. So maybe we can have a broader discussion of that after everyone's presented their data access.
Yeah, cool. We have a couple more slides, so I'm going to go through them pretty quickly.
There is one question from Vladimir about whether or not there's a learning portal for the eDNA Explorer to learn about all these tools.
Great question. So there's a really extensive FAQ. And so if you go to resources, there's like some docs that walk through in a pretty accessible way, I hope, all of the like, how do you do taxonomy assignment? What are your reference databases? Like, what does this mean? And we also have a chatbot that is pretty good on eDNA Explorer, so you can ask it to help you interpret your graphs. There's tooltips on the eDNA Explorer. If you kind of hover over things, you'll see little tooltips that unpack some of the like statistics and different things. And we are always happy to Zoom with you and walk you through the website for anyone who wants to get more details. Also, you don't have to be a contributor to download the data. I believe right now you have to be a contributor to access the ASVs because those are many gig files, but or we're thinking about that, but we're trying to make this like fully open. So if you wouldn't mind testing it out, I think everyone can download the ASVs and everyone can download the data, the taxonomy tables, and that also has raw and decontaminated. So if you want to do your own processing, you can do that on your own. The ASV tables are large and have a lot of steps, but everything's explained in the resources.
Thanks.
Cool.
And then one more question about if we were to have a private project, would all the features where you can compare samples across the project still be available?
Yes.
Okay.
Yes, and that's with an umbrella project. You would basically click all the other projects that you want included in your modeling, plus your private project. So that's like, we're working on it really, really hard right now, hopefully available in a couple of weeks.
Okay.
And I'm going to say with like, if we look around Eden Explorer's results, there's like really great fungi data. We think we do a really good job for 16S, and CO1 is underperforming. So we're really trying to test out, like, if we add the bold reference database, how much do we gain? If we do some deep learning assignment methods like using ProTax, what do we gain? So I think we do a decent job, but not to like the species level resolution that you're looking for. We only have a couple of hundred invertebrates that are identified to species level, and a lot else is clustered up to genus level or higher. And right now we're actually like throwing away like 60% of the data, 70% of the data based on our filters. So we're being too stringent. So I apologize. The data are all available, but we really need to improve our CO1 approach. So you can check out the data here. Your insights would be really valuable as we work to resolve this. In the next couple of weeks, hopefully we'll have an alternative that is really attractive for everybody. You can see the distribution of like, we have like 100 families from CO1 that we've been finding, but like we expect way more species. Some groups like Diptera are particularly high in reed counts, but low in species, the percent of reeds that have been assigned to species. So Diptera are sort of like the worst contenders. But we have some groups that are like really, really low species assignments. So you can see this graph here of the percent of reeds that have species level assignments across some of the major orders. And then we do better for the other taxa. And so when we say taxon lineage, that's any level that it's resolved to from phylum to species. And we really want species level assignments because we want to be able to make species distribution models, habitat suitability models, and like help you forecast like if you think that that species is going to be there, especially with the environmental variables, you should be able to predict how those species will fare under climate change and under land use change and all sorts of really cool things. And so we want you to have that capacity on Eden Explorer. So for the past, we took CalATBI data from 2,000 samples that were before the CIB processing that Julian and Ajith were doing. And we did gradient boosted random forest to make wall-to-wall maps of dominant, like the major, like if you were to sort all biodiversity into like 19 clusters. This was Levi's work that he led. And then to look at the richness patterns. So we have those maps available. And those are through the CalATBI site, not the Eden Explorer site. And you can just go under Explore Data and you'll find the link to those. It's in a paper. And then with the new samples, we want to be able to reprocess everything, include the new bold data. So with 1,000 more samples and newer reference data, hopefully we'll get way, way, way better species identifications and predictions. But already we're like not bad at predicting community types, even with like less than perfect data. We get really high accuracy compared to all the random forest work before. And so like I just wanted to shout out like the R-squared for fungi is like above 0.7. This is cross-validated. So we can really tell you what environmental layers we have over 380 variables are underpinning the beta diversity or the community composition. And that's weaker for 16S, but like it's greenness and drought predictors for fungi. It's a lot of human influence for CO1, and it's a lot of soil characteristics for 16S. And that's pretty cool. And then just to end on a couple of things that people have asked. So topsoil versus 10 centimeter depth. Are you ready? There's more inverts at the top by a little bit more. So good that you made that choice as one to pick. And we did not find significant differences in richness or evenness or composition between topsoil and 10 centimeters depth. So the site signature is really available in both, and the topsoil appears to be pretty well mixed. And this trend holds true for other markers. And then people have also asked, what about ITS1 versus ITS2? Is it worth doing both? And the answer is yes, it is. We see very much unique taxa in ITS1 and ITS2. ITS1 only, there are 4,000 taxa that are unique. ITS2 only, there are 1,000 taxa that are unique. ITS1 does way better at species level assignments than ITS2. And it looks like ITS1 is slightly better for Basidiomycota, Anaascomycota, and ITS2 ends up getting more plants that are sort of off target. And so if you want to sign up and have a profile and start to like organize your projects and also upload your own data, you are welcome to just create a login. And if you go to the search bar or Explorer, you can find the CIB plates just by looking up California Institute for Biodiversity or plate and find the plate number. You can message the project owner if you want to be added. They can add you a contributor, but anyone who's a contributor can also add you. So once you're a contributor, you can also add other people. All right, that's it. Thank you.
Awesome. Thank you so much. And if people on the call are wondering how to figure out what your sample IDs are, we can send those to you. We have those available on our dashboard where you can easily download all of the metadata that you collected during the collection of the samples. So you can link your sample data back to these samples. So with that, I'm going to pass it over to Austin, who's going to talk a little bit about insects and what's available on Bold.
Hello, everybody. For those who haven't met me, my name is Austin Baker. I've been leading the insect new collections sort of organization. Let me share my screen here. There we go. Okay, can people see my presentation?
Yep.
Okay. So I'm going to lead you through the easiest way to access the insect data at this time. It will get easier in the future, and I'll go over the reasons why that is. But right now, the easiest way is to start at data 1, where the complete project backup has been uploaded. And from there, you can download all of the data. Bold is the primary database where everything is kept up to date in terms of taxonomy. It's got all the specimen information, sequences, and images. And there are some ways to use Bold from a public perspective that are easier than the workbench, but I'll go over both. And then the last way to access the data is from the Google Drive called CalATBI New Collections. And this you need to ask for permission to get added to the project. And I generally don't recommend this for most people unless you want old data backups or protocols or things like that. So from the beginning at data 1, I recommend this for most users right now. The data is not entirely released on Bold. When I say that, I mean most of the specimens are publicly available, but the data set altogether as one like tagged data set is not going to be available until the publication I'm working on is finished, which should be in about a month, pending additional revisions, but it should be soon, ready soon. So from data 1, you want to navigate to portals and then all taxa biodiversity inventory. What that looks like is on this page, we're going to go to portals here at the top. And then the all taxa biodiversity inventory. And from there, you scroll down, and the California Insect Barcoding Initiative is the second link here. Might not always be the second as more things are uploaded, but it's going to be called the California Insect Barcoding Initiative. And from there, you can download the entire spreadsheet that has all of the insect data. That spreadsheet looks like this. So it has your Bold sample IDs, your bins, which are essentially your molecular species identifications, as well as however much actual taxonomy has been added to the specimens. It's got all the metadata in terms of when, where, how, and who collected it. And from here, you can actually go onto Bold and use that information to search for specimens individually. So this slide just sort of going over what I just showed, how to access that spreadsheet. The important fields here are going to be sample ID and the bin in terms of how you can search for things on Bold. I'm using this specimen as an example. So the Bold portal, which is separate from the Bold workbench, is a way that public can access the data very easily. The Bold workbench has more tools, and I'll go over that in a little bit, but it also requires permission and an improved account to be able to access it. So from the Bold portal, you can take a specimen ID like this. And this is what the Bold portal looks like. You can enter all sorts of search terms, but in this case, if we're searching for specimens from the project, we want to enter the specimen ID. And then in brackets, it'll add IDS, which means that's an ID. And it should take us immediately to the specimen, including the image. If it loads, there we go. Where it was collected, all of the metadata, everything like that can be found on the specimen page. This also includes the sequence information, pretty much everything you'll need to know for each specimen record of the 1 million+ specimens we've generated so far. You can also search for an entire species by searching the bin, and this will bring up multiple specimen records. But whenever you bring up multiple records, you can also bring up records that are from outside the project. For most taxonomists and things like that, it's not going to matter. But if you're really curious on just things from the project, you need to be careful about how you search. Okay, this is just going over what I just showed. The Bold workbench, I don't recommend for most users, but for people who are taxonomic specialists that need access to the entire data set before it's released to the public, what you have to do is make an account with Bold, email me with your account name, and then I will email the Bold admin and get you added to the project. So it takes several steps, which is why I don't generally recommend it. It'll show you, the Bold workbench has a lot of really cool tools that you can use. If I click on one of these projects, which are all sub-projects within the insect barcoding initiative, it gives you the option to download the sequence data or the data spreadsheets, which include all the specimen data. You can analyze the data, build neighbor joining trees, do whatever you might need to do to figure out how those sequences are related to each other. So there's a lot of really great tools in here, but for the average user, that's generally not necessary. And then the last way is to access through the Google Drive. Again, I don't recommend this for most users unless you want previous data backups, old meeting notes, sampling protocols, things like that. This is where we've kept a record of all of our insect barcoding initiative documents. And then you just need to email me, ask for permission to access the Google Drive, and I'll send you a link. And within the drive, we have a variety of folders, including our Bold data backups, which go all the way back to February 2024. And the most recent, I believe, is from November of this year. And so that's the easiest way to access the insect data at this point. Once my publication is finished, Bold will release the insect data as a project, so you can access it all in one place on Bold, and that will then become the easiest way to access the entire data set.
We have one question about how are potential new insect species determined?
So the initial determination is dependent on a molecular ID. They use the DNA barcoding sequence to cluster things that are potentially species, and a lot of those don't have species names put to them. They might just be identified to the family, subfamily, or genus level. And cases of like a lot of the fly species, those are going to be brand new undescribed species. But you need to have a pretty good knowledge of what's out there. You need to be a taxonomist to go through the pictures and be able to identify, what is and is not a new species. If you do want access to the specimens, also you can reach out to Bold, and they do specimen loans from this project. And so far, I've approved every loan that's allowable for us to send out. So taxonomists can then get the specimens and improve identifications, because Bold is sort of this iterative system that's constantly improving in terms of the taxonomy of our specimens. Any other questions? Okay, thank you.
Awesome, thanks so much. That sort of concludes the data sharing portion for the people online who are help with the sample collection. If you guys like, you can stay on. We're kind of doing more business as usual with the core ATBI members. Again, you're welcome to stay on. I'm going to stop the recording so we can use this part to send to other partners as needed.